Hello everyone, we are going to discuss what makes actually stem cells unique, the uniqueness of stem cells. At the beginning of embryonic development, stem cells undergo symmetrical cell division. They divide symmetrical, where one cell splits and give rises to two identical cells that have the same potential. This is why we said the stem cells in the early embryo remain the same and are identical. Interestingly, this is also how fully mature cells in your body divide. What is the technical term for this that you guys probably learned in biology? Mitosis. Then at blastocyst formation and gastrulation, the stem cells start to divide asymmetrically. When these stem cells divide, they give rise to two cells that are different from each other. One of the cells remains a stem cells, the yellow one, and the other changes into a progenitor cells, the green square. That progenitor cell is a young cell that will change into a mature cell type like the epithelial cell in green. Asymmetrical cell division is when a stem cell divides to produce two cells that are different from each other. One of the cells remain a stem cell demonstrating self-renewal, and the other differentiate into a progenitor cells. Okay, so that stem cell divided to produce a green progenitor cell which differentiate into a green skin cell. That stem cell can divide again, this time producing a different type of progenitor cell which matures into yet another cell type like this brain cell. Okay, so what makes a stem cell unique? First, it maintains its own population, self-renewal, and secondly, its capacity to give rise to different progenitors that change into a mature cell differentiation. So what do stem cells look like? There are thousands of human embryonic stem cells growing in this circular colony. The human embryonic stem cells are grown on top of mouse feeder cells, the elongated cells, which provide essential nutrients. When we culture embryonic stem cells, we are trying to stop the majority of the colony from differentiating so that we can maintain a pure population of self-renewing embryonic stem cells. Why do you think this would be important? We want them proliferating, not differentiating. So we have a continuing supply for research purposes. Scientists can label embryonic stem cells using antibodies attached to fluorescent molecules. On the left, the smaller blue dots in the green area label the nuclei of the embryonic stem cells, and the blue dots outside the colony label fibroblast feeder cells. Why do you think fluorescent labeling is important? This is how scientists can keep track of which cells are stem cells and which are not. Embryonic stem cells like to grow in little colonies. On the right side you can see, spaced out over the disc instead of in one big layer.